All, this is Dr. Mubin Sayed from FLCCC. Today I am here with one more episode of the Long Story Short with Dr. Mubin Sayed. And today we'll continue our discussion about the chronic inflammation. Chronic inflammation is a situation which is present in more than 50% of the population. I think in the US, 60% or more population has some disease that is related to chronic inflammation. Worldwide, more than 50% of the deaths annually that occur are because of some disease that has a contribution of chronic inflammation. And nowadays, long COVID and the vaccine injuries are also looking like chronic inflammations. So it is important to understand chronic inflammation. The discussion that we are going to do today is about the regulation of inflammation. And of course, when you are listening to the discussion, keep in mind that if any of these mechanisms, when they break down, then we are not able to regulate the inflammation anymore and we end up in chronic inflammation. The next episode is going to become even more important, and today's episode is going to create a service for that one. And the next episode is going to be T regulatory cells. So let's start our discussion. So the regulation of the immune response, first of all, realize that the regulation versus suppression are two different things. Regulation of the immune response means to increase the immune response activity when needed and to reduce that activity when needed, to stop that activity when needed. That all is called regulation. So it may be upregulation, downregulation, or homeostasis. On the other hand, inhibition or suppression actually means to stop the immune system from doing what it is doing or to suppress the immune system from doing its function. Now, if we look at this diagram in here, look at the two central boxes. This box here is the representation of the innate arm of the immune system. This box over here is the representation of acquired arm of the immune system. Immune system is divided in these two arms. And normally what happens is that when we have an allergen or a pathogen or an antigen, our innate arm takes the first go. It becomes active first. And as it starts fighting with that allergen or pathogen or whatever antigen it is, it also starts activating the acquired arm. In turn, when the acquired arm becomes active, that would then activate or amplify the innate arm, and this process will continue where both arms will strengthen each other's function, and that is how the inflammation will be created and sustained. On the other hand, when these mechanisms start, when these activities start, various cells and proteins that are participating in inflammation, there are going to be levers within this mechanism that would try to control, reduce, suppress, and stop the inflammation as well. We do not want the inflammation to continue forever. So controlling mechanisms will kick in at the time when the activating mechanisms have started. Many of the controlling mechanisms actually have a time attached to them. That is, they kick in right away, but they start doing their action within a few days. This is why inflammation starts, does its job, and after a few days it stops. But there are many other factors for the inflammation to stop as well, and we'll look at that. Now, for chronic inflammation, these controlling mechanisms that we are studying and will study today, these could become excessive. And that control could be an upregulation, and excessive upregulation could mean chronic inflammation or more intense inflammation in the acute phase. Then the controlling mechanisms are also necessary to prevent an attack on the body, on our own body. So our own immune system attacking our own body, we have to prevent that. And these controlling mechanisms help with that. This is called self-tolerance. And if that breaks down, we would then also end up in chronic inflammation with autoimmune diseases. In addition to that, the inflammation is regulated and controlled over time and then various locations. For example, it is possible that inflammation is going on in one part of the body and the other parts are making sure that it does not spill there. Or in some cases, it does spill there and causes the symptoms. Similarly, the intensity of the inflammation is controlled as well. The cytokines. Cytokines are, as I discussed before, these are the chemical molecules that are released by the immune cells. These are actually released by many other cells that are present at the site of injury and inflammation. And these cytokines in turn are going to help control. So controlling mechanisms are not only cell-based, but also protein-based. Now let's look at why some of these mechanisms, the controlling mechanisms, fail, or why do we end up in chronic inflammation. One such example or cause could be that whatever is the environmental factor, maybe that is an allergen, or maybe that is a pathogen, or maybe that is a body tissue that our immune system has started thinking it is a foreign tissue. Whatever is the antigen or the triggering factor, that just continues to be present. And if it is continuously present, then our immune system, in most cases, would continue to attack it. In some cases, we develop tolerance and energy, ANERGY. In some cases, our cells develop tolerance and energy, but in some cases, they do not. And when they do not develop this tolerance, then there is going to be an attack on the tissues. So pathogens continuously present, for example, mold continuously present, fungi, viruses, bacteria. Then the breakdown of the self-tolerance. Then when the inflammation starts, the receptors on various immune system cells, for example, macrophages or dendritic cells or neutrophils or natural killer cells or other cells, B cells, T cells, many times when the receptors on these cells are activated, these receptors internally start processes and mechanisms that would help these cells try to calm down the same cells. They would try to calm down themselves. This is like if we become angry and do a self-talk to say, all right, calm down, slow down, no worries, relax. Similarly, these cells can actually release molecules that would act on the same cells and calm them down. These can produce molecules that would act on the neighboring cells and calm them down, even remote cells, and modulate their activity. So triggering event and the receptors that are triggered can in turn start the inhibitory activities, and I'll give you an example of that. Then immune system can also become modulated and controlled with time. This is why many times you would have heard, for example, in vaccine injuries, that as the time passes, patient is generally becoming better and better. That may be because the antigen is being removed. That also may be because the cells that are involved, they slowly come to grips with it. They slowly start dying out or clearing out or reducing their activity, developing energy and becoming less active. Then on the time basis, there are actually regulatory cells and regulatory mechanisms that do not become active right away. For example, let's say there is a tissue damage. Let's say my deltoid muscle got vaccine, or there is some other injury to some other part of my body. The immune cells in those areas, they started becoming active right away, innate arm cells. They started inflammation or a response. Now, if the regulatory mechanism or inhibitory mechanism becomes active right away, regulatory mechanism means upregulation or downregulation. So that doesn't matter, that would become active right away. Inhibitory mechanism, if that becomes active right away, then we would actually not get the inflammation because we just started it and stopped it. So in such cases, the cells that are going to regulate this inflammation are activated, but they would take time to create their effect. And that gives a window to the inflammatory cells to do their activity and then start slowing down and calming down, which we would in medical terms say go back to homeostasis or normal state. There are many other regulatory factors, many other. The whole immune system, every cell and protein is involved in some sort of control and regulation in addition to activity. So some examples are tolerance development, exhaustion of the cell. If you repeatedly expose a cell to some antigen, it can become exhausted. There is retraining of the innate arm cells. There are genetic factors that can be playing a role in immune system's behavior and tolerance or incorrect tolerance. No production of the immune response to production of extra immune response and chronic inflammations. Then the memory cells. This is a very important thing to keep in mind. It is possible that sometimes in the beginning of our life, we may not be reacting to certain things. But as we age and we accumulate the memory cells that have responded to various antigens, those memory cells are present with us. And the more the memory cells are present, and if they are active, the more reaction we will do and more allergies and inflammations we can get. And then mimicry. Sometimes our body or our immune system actually does not attack our body. It is fine with the body and the tissues, or the self-antigens as we call them. And then some event occurs, some allergen, some drug, some bacteria, virus, fungus that arrives in our body and creates a molecule that looks to our immune system as a foreign or a negative thing, although it is part of our tissue. So our existing tissue that immune system did not react to would suddenly start looking like foreign material or an offensive agent. Our immune system would start then attacking it. And that is an example of molecular mimicry, or the mechanism of molecular mimicry. So with this, as we go into the detail of various regulatory mechanisms, let's just have a very quick overview, high-level overview of the immune system, so that when we are talking about various cells and immune system cytokines, we are aware where we are talking. So this part is the innate arm. Innate arm, dendritic cells, macrophages, neutrophils, NK cells, many other kind of cells are being discovered nowadays. So innate arm, first responder. Innate arm in turn activates the acquired arm. Acquired arm is made up of helper cells, which start with a naive T cell that connects with an antigen presenting cell. Then the naive T cell, in the presence of interleukin 4 and absence of interleukin 12, becomes a T helper 2 cell. T helper 2 cell, which is also a helper cell, would then in turn cause a B cell to become a plasma cell. And plasma cell is nothing but an active B cell making antibodies. A naive T cell in the presence of interleukin 12, and please keep interleukin 12 in your mind, because somewhere down the road, today we'll talk about interleukin 12, interferon gamma axis. So interleukin 12 is usually generated by or produced by the innate arm cells. So interleukin 12 is present that can convert a naive T cell to a T helper 1 cell. T helper 1 cell would in turn activate the cytotoxic T cell, and a cytotoxic response would occur from the acquired arm. In addition to that, there are possibilities that a naive T cell can become T helper 17, or it could become another cell class which is called T regulatory cell. There are many phenotypes. Phenotypes mean many markers of the similarly functioning cell, for example, T regulatory cell. However, that T regulatory cell may have various kind of markers on it, for example, CD25 and IL-2 receptors, which are also the CD25 receptors, the same thing, and so on. So other receptors are present as well. So we'll see those in the next lecture. So next lecture, we'll talk about regulatory cell. Today, we'll talk about these mechanisms overall and how various cytokines help. So in a healthy tissue, in a healthy body, if an immune system cell, let's say this is a dendritic cell or a macrophage, connects with a healthy tissue, let's say this tissue is healthy because it is not infected by a virus, bacteria, or fungus. It is not cancerous. It is a normal, healthy, functioning cell. If that is happening, then what happens is that mostly healthy tissue cells will have some sort of a molecule on them that would inhibit the engaging innate arm cells or even acquired arm cell. So this is an example of a regulatory mechanism. For example, B7 is an example of such mechanism, and CTLA4 is another example. The point here is healthy tissue may have molecules on its surface that would stop the attacking immune system cells. On the other hand, when a healthy tissue is not healthy anymore, it has become infected or it has become cancerous, and now this tissue is not able to create extra molecules on its surface because the cells are sick. They cannot make it. If they cannot make those extra molecules that are inhibitory molecules, then what happens is when an immune system cell, for example, let's say a macrophage or a dendritic cell, engages with this unhealthy tissue, and unhealthy tissue is not able to stop it by engaging with those extra molecules, let's say B7, then the result is that this innate arm cell will become active. Even acquired arm cell can become active, and that activity would then start inflammation over here. Now imagine if for some reason the immune system cell is actually being engaged for the inhibitory molecule, but the cell is not responding to it. We'll end up with the chronic inflammation. Or imagine if the healthy tissue cell actually has the molecule to stop the immune system cell, but that molecule itself is not in correct shape or correct structure, or is not functioning correctly. There is a genetic problem. So even when the engagement of inhibitory system is there, it is actually not functioning correctly. And even then, in that case as well, we'll end up with the chronic inflammation. Then some more examples. These are examples of how the inflammatory control systems can break down. So interleukin 10, this is a very important interleukin to keep in mind. Interleukin 10 is released by many cells, most importantly T regulatory cells and the innate arm cells and T helper 2 cells. These are very important cells to keep in mind for interleukin 10. Interleukin 10, when it is released, what it does is it acts on the innate arm. So here is a delicate point. Stay with me for a second. Let's start from the innate arm. Here is the innate arm cell, let's say a dendritic cell. It has become active because it found some offensive agent, some antigen. While it is becoming active and starting the acquired arms activity here by this engagement, at the same time, innate arm cell is also releasing interleukin 10 that is acting on the same cell and asking that cell to calm down, slow down, stop the response. So this is an example of negative feedback. Negative feedback is that I am telling myself to calm down. It's not negative or a bad thing. It is negative that the function is being reduced. So this is an example of a control, inhibitory control of the innate arm on the innate arm to reduce the activity. Why are we doing this? Why is our immune system keep doing such things? Our immune system does not want our inflammatory systems to run away, to become runaway systems. They don't want it to become chronic. So with every step that is activated, there is a controlling mechanism that is started. So innate arm produces interleukin 10. Then T helper 2 cells that are going to activate the B cells, and B cells in turn will make antibodies. So that is called the humoral response of the immune system, correct? T helper 2 cell produce interleukin 10 as well, which in turn, so remember this is acquired arm, these in turn cause suppression or inhibition or reduction in activity of the innate arm. So here you're seeing an example of the acquired arm reducing the activity of the innate arm. And then the most important thing, T regulatory cells also produce interleukin 10. We'll talk about T regulatory cell in the next lecture. This interleukin 10 that is produced is also going to act on the innate arm to reduce the action of the innate arm. And now you can see a breakdown in the whole interleukin 10 activity can cause chronic inflammation. For example, let's say if the dendritic cell or macrophages do not respond well to IL-10, or if the IL-10 is not produced correctly, then we can end up with the chronic inflammations. That also means IL-10 can be a therapeutic intervention as well. Then continuing, now here is another example. This is the example of the innate arm cell. So imagine this is a dendritic cell or a macrophage. These are the first responders. So start from here. Imagine we have a dendritic cell. This dendritic cell on its surface has Toll-like receptors, as we've been talking about. Toll-like receptors themselves are further classified, for example, in DAMPs and PAMPs or so on, the pattern matching receptors, or receptors that would recognize something in the environment that is not right. A virus or a bacteria or a fungus or an allergen or a cancerous cell or a broken cell, whatever it is. Now, we talked about it that TLR receptor activation is really important to start the whole process of inflammation. But do you know that as the TLR receptors are activated to activate the immune system to cause inflammation, simultaneously within these cells, there is another set of proteins that are activated. And remember, it takes a few days for these proteins to become more active. So it's not that the macrophage just became active and within 1 second it turned itself off. Then there is no benefit of this macrophage. So there is always a balance. So here, toll-like receptors are activated. That makes this macrophage or dendritic cell active and take part in immune system inflammation. At the same time, within the same cell, there is a protein, or actually a cluster of proteins, which are called collectively suppressors of cytokine signaling, or SOCs. Suppressors of cytokine signaling. What does that mean? If we forget about these proteins for a second and understand what's going to happen with this cell. As the cell becomes active, it is going to release cytokines, chemical molecules, to tell the neighboring cells to become active. Those cells that will become active in turn will produce cytokines as well, which will work on other cells plus themselves plus this cell. Now this cell is producing cytokines that would also work on the same cell, as I discussed before. So the end result is that an active innate arm cell will have receptors on its surface that will respond to cytokines, and those cytokines would regulate its function, mostly upregulating it. Now we don't want that to continue to happen, because if that continuously happen, then we would end up with chronic inflammation or more intense inflammation. So these suppressors of cytokine signaling proteins that are simultaneously activated, they will go and bind with the second messenger system of cytokine receptors. What does that mean? Cytokine receptor is on the outside of the cell. So imagine if I am a cell and I have this little hand is my receptor on my surface with which a cytokine can come and bind and activate me. Or if I'm already active, then increase my function or sustain my function. Now this receptor that is on my surface is internally connected on the inside if I increase the size here. So this is the intracellular, or inside the membrane of the cell. There are more proteins connected to this receptor. These proteins are the second messenger system. For example, in case of cytokine receptors, many of them have JAK-STAT system or Janus kinase system. This JAK-STAT system that is on the inner side will in turn activate many of the actions within the cell. So that's a good thing. We need the cell to respond to cytokines and do various activities as a result of those signals. But we also want the cell to at some point reduce its activity and calm down. So these SOCs molecules that have become activated, their job is to suppress the JAK-STAT mechanism's action, which will in turn suppress the activity of this cell, which will in turn inhibit the action of this cell. Isn't that a wonderful mechanism? So of course this also means that SOCs are molecules that can be looked at for therapeutic interventions to say, can we give something that can help reduce the cytokine responses by a cell? So this is another example of inhibitory control of the immune system that is built into it. And if this control is somehow broken down, then we'll end up with the chronic inflammation. Either broken down or overwhelmed. Overwhelmed would mean what? That these proteins are active. They are trying to do their work. But there is just so much bombardment of the cytokines. And there are just so many receptors that are active that these proteins and their inhibitory process is overwhelmed. Okay, so continuing. One important takeaway that you may be seeing is that for every cell and every action of the immune system, there is a controlling mechanism as well, which may be upregulating or downregulating. So then interferon gamma. Remember I said in the beginning of the talk that keep in mind interleukin 12 and interferon gamma. Interferon gamma is another molecule which is very important target of many therapies for immune disorders. Interferon gamma is mostly produced by the acquired arm of the innate system, especially the cytotoxic arm. So what is the role of IL-12 and interferon gamma? When the innate arm becomes active, let's say a macrophage or a dendritic cell, they bind with the naive T cell to activate them. We saw that before as well. At the same time, they can produce interleukin 12. If the interleukin 12 is produced, then this naive T cell will become T helper 1, which would then activate the cytotoxic T cell. Now, T helper 1 also produces interferon gamma, which in turn acts on the innate arm cell and amplifies them. We saw interleukin 10 that suppresses them. Here we are seeing interferon gamma that amplifies the activity of the innate system. Now, when the interferon gamma is released by, for example, acquired arm, or interferon gamma can also be released by the innate arm cell. For example, natural killer cells are part of the innate arm and they produce a lot of interferon gamma. When that interferon gamma is present, it's a cytokine, correct? It is going to now act on dendritic cell or macrophage, and it upregulates the activity of the macrophage. It increases the major histocompatibility complex production of the macrophage, which is this little molecule on which an antigen is presented to the other cells. So here you're seeing a molecule that would actually enhance the activity. And now imagine if there is too much interferon gamma, that would mean there is too much activity. That could become a bad acute situation, or if this activity sustains, then that would become chronic. Now, remember, this is a cytokine that is going to enhance the activity. If we go back here for 1 second, now I think you could realize how important for these SOX proteins to become active, because interferon gamma is going to come in and attack here or attach here and cause the activity. And if we don't have SOX, then the interferon gamma would have uninhibited effect of upregulating the system. So do you see how these things have to work in a balance? And any shift in balance would become a problem. And of course, then the external intervention, the therapies, will have to compensate for these breakdowns. And the final example for today, I think you would really enjoy this example. This is the control of the antibody production. And this mechanism that I'm going to discuss is called antibody feedback mechanism. And overall, before we look at this diagram, overall, what this mechanism is, that when a B cell produces an antibody, that antibody, when it connects with an antigen, that complex, antibody and antigen complex, can in turn switch off or reduce the activity of this B cell. This is an example of, again, negative feedback, where the output of a cell is causing a negative effect on the same cell to reduce the output or to reduce the function. So how does this happen? Beautiful mechanism. How does this happen? So let's start from here. Number 1, we have some pathogen. Let's say this is a spike protein from a vaccine, or let's say this is a SARS-CoV-2 or some other streptococcus, staphylococcus, something. It gets attached, or B cell recognizes that antigen, and B cell receptors are IgD and IgM. So B cell connects with them, recognizes them. Then B cell needs co-stimulation from T helper cells. We have seen those mechanisms earlier. Eventually, the B cell becomes active. When the B cell becomes active, it produces a lot of antibodies. Those antibodies are released out in the tissues, and from there in the blood, and then it would go everywhere in the body, the antibodies. Now, the antibodies, what is their function? The antibodies' function is to connect with this offending agent or the antigen. So, for example, to spike protein or to SARS-CoV-2 proteins or some other antigens. Now, this antigen-antibody complex, this bound together thing, complex, we call it antigen-antibody complex, it will have its own biological outcomes. But on the same cell, there is an interesting outcome, and that is that this complex, if you see here on number 3, this complex can now reattach or can be recaptured by the same B cell. And how does that happen? This is a beautiful mechanism. I'm going to increase the size a little to show you. On the B cells, we have a receptor for antibodies or for IgGs. The receptor is called fraction constant IgG2B receptor. What does that mean? On an antibody, so if we see here this red thing, an antibody has 2 main parts. Variable part with which it binds to the pathogen, and a constant part which is constant. The constant part is also called FC, fraction constant. And on the B cell, there is a receptor that can bind with the fraction constant part. So now imagine we have an antibody that has captured a pathogen or some antigen. That complex gets attached on the surface of the B cell with the Fc-gamma receptor. This fraction constant, gamma standing for IgG, 2, and B receptor standing for a receptor on the B cell. So the whole name Fc-gamma R2B receptor is going to attach with the complex. Then at the same time, this complex has of course an antigen on it. That antigen will bind with the B cell receptor for the antigen, which is IgD and IgM. So there is another IgD and IgM that is also bound to this pathogen. This short circuiting in turn tells the B cell that, hey, you are able to work. Hey, B cell, you are able to work. Your work is successful. The antibodies you produced have captured the antigens, and we can actually see them attached here on our cells. This also means, hey, B cell, now it is time to reduce your function. And how does that happen? So this Fc-gamma, I'm going to increase the zoom level once again. This Fc-gamma receptor has a cytoplasmic tail, or a part that is inside the cell. So once again, if you look at my head, there is a receptor on my head, and then there is a part that is inside the skull. So that inside tail has a tiny protein attached with it, which is called ITIM. Now that ITIM has another little protein attached with it, which is called SHIP, or SH2 domain containing inositol phosphatase. But simply let's just call it SHIP, another protein. When this whole thing, this whole cluster of activity occurs, and it is necessary that Fc-gamma is connected and the IgM or IgD is connected. Once this whole cluster is activated, then SHIP is activated by ITIM. SHIP in turn, it is an inositol phosphatase. So it is an enzyme that will go and reduce the activity of another enzyme that is present in the membrane of the B cell, which is called phosphatidyl inositol triphosphate, or PIP3 enzyme. Now PIP3 enzyme's function is to work with the B cell receptors, IgD and IgM, and activate the B cell. So if you suppress the PIP3, then you would, even when there is antigen attached, you will still reduce the response of the B cell. So in summary, what did we just end up with? We have a B cell that responded to an antigen, and it produced the antibodies. Those complexes of antigen antibody then in turn reconnected to this B cell, and their reconnection confirmed to B cell that the function is done, my work here is done, and in return, inside mechanism started to reduce the B cell's response to this antigen. So even when that antigen is now present, even when it is connecting, even then B cell is responding less. That is control. That is inhibitory control. It does not want to go mad with producing too much antibodies. Now think about it. If this mechanism isn't working correctly, will B cell be able to turn itself off, or at least reduce its activity? No. Will we start ending up with the continuous production of antibodies? Yes. Will those antibodies then would cause chronic inflammations? Yes. So again, the point was to show various controlling mechanisms. Thank you very much for listening to this. In our next discussion, we'll do a crucial part of understanding chronic inflammations and understanding how to control the chronic inflammations, and that is T regulatory cells. So today, this is the discussion. Hopefully, I'll see you soon with the next episode. Bye-bye for now.